Hey guys, it's Hunter and Ann. Today we'll be discussing the effects of strawberry and ginger polyphenols on human leukemia 60 cells. Gingerol, which is an ingredient found in the rhizomes of ginger, has been shown to have strong anti-inflammatory activity, which is considered closely associated with its chemopreventative potential. Gingerol is also found to inhibit the viability and DNA synthesis of human promyelocytic leukemia cells. The cytotoxic effects were associated with apoptosis cell death.
Strawberries are rich in bioactive compounds such as anthocyanins and flavonols, and they have also been found to have anti-inflammatory effects on the body. Due to this, strawberry polyphenols are being explored as possible treatments for various types of cancer, especially breast cancer. They have been found to reduce viability and induce cytotoxicity in breast cancer cells. In an experiment done on mice, strawberry polyphenols were fed to mice with breast adenocarcinoma tumors. With no other treatment, the tumors were gone by the conclusion of the study. In this study, strawberry and ginger polyphenols were used to treat HL60 cells. HL60 cells are formed when a blood stem cell gets stuck in the myeloblast phase. This leads to constant cell division and no further cell differentiation.
The preferred way of killing HL60 cells is through apoptosis. Apoptosis causes shrinkage, fragmentation, and blebbing of the cell. The final apoptotic bodies are then usually engulfed by phagocytes and do not cause inflammatory reactions. In the viability portion of our experiment, treated and control cells were placed in WST8. When viable cells are present, their NADH will convert the WST8 to a yellowish color. We can then measure the absorbance at 450 nanometers to determine the viability. The less absorbance, the less viable cells, showing that death of apoptosis or necrosis is occurring.
The first two columns of this figure represent our control groups. The first control was media and cells, and the second control was media, cells, and 70% ethanol. The treated cells were compared to the second control. While the strawberry treated cells did have a slightly lower absorbance, this difference was considered insignificant. The HL60 cells treated with ginger had a much lower absorbance of only 0.2 nanometers. This was considered statistically significant compared to the controls, meaning that ginger resulted in a much higher death rate of the HL60 cells.
By looking at cells dyed with cyto16 under a confocal fluorescent microscope, we can visualize the nucleus of each cell. We are capable of doing this because cyto16 binds to the DNA within the cells. This allows us to determine the morphology of our cells and determine whether their death was due to apoptosis or necrosis.
As gone over in the beginning of this video, the stages of apoptosis are nuclear condensation, fragmentation, and blebbing. This process results in no inflammation because the plasma membrane remains intact. In the morphology of the ethanol-treated cells, only a few points of nuclear condensation are seen, and no further apoptosis is seen. In the positive control of camptothecin-treated cells, the morphology assay reveals condensation as well as fragmentation. There are also far fewer cells in this control, showing that many have already been killed through apoptosis. This image shows the morphology of HL60 cells 24 hours after they were treated with strawberry polyphenols. There are some instances of minor condensation of the nucleus, but no further apoptotic steps are seen. This treatment caused similar results to the ethanol control, revealing very little effect of the strawberry treatment.
After 24 hours of treatment by ginger extract, there is clear signs of condensation, blebbing, and fragmentation. This would suggest that ginger is a strong inducer of apoptosis within HL60 cells. Oxidative stress detection reagent green is a cell-permeable dye that binds to ROS and fluoresces green. It is apparent by the fluorescent green that ginger treatment did cause reactive oxygen species which is detrimental to the cell and can cause DNA damage.
Another way to determine if a cell has been damaged is through the COMET assay. By placing treated cells in agarose on a COMET slide, DNA damage can be visualized. After performing the COMET assay, different types of COMET tails can be seen. The length of the tail indicates damage to the DNA. This is because coiled, intact DNA moves slowly on the COMET slide, while fragmented DNA moves quickly, resulting in a longer tail. Tails are given a score from 0 to 3, with 0 showing no tail and 3 showing a long and spread out tail. In the positive camptothecin control, levels 0, 1, and 2 were all seen. The majority of cells seen are level 0 and show no tails. This shows that the DNA of these cells has not had a significant amount of damage. Unfortunately, COMET assays performed on both ginger and strawberry treated cells did not yield good visual results. In the case of ginger, it is likely that the cells had been too destroyed with very little DNA remaining to see results.
In conclusion, when it comes to strawberry and ginger as tools to effectively kill leukemia cells, our study suggests that ginger is in fact capable of decreasing viability, causing reactive oxygen species, and inducing apoptosis. Whereas with strawberry, we can't say it significantly lowers viability, it definitely didn't cause ROS, and it didn't show any signs of apoptotic blebbing fragmentation. However, there was some signs of condensation.
This experiment showed that strawberry did not have any significant impact on HL60 cells. It was unable to induce apoptosis or reduce viability. There are two possible ways to take this experiment next. The first is to continue using strawberry polyphenols on HL60 cells. Perhaps larger amounts of strawberry would have an effect. I would recommend trying multiple treatments over a longer period of time and then repeating the tests. Another direction would be to just try using them on a different type of cancer cell. Strawberry was found to be effective against breast cancer cells, so it may just be a matter of finding a cell type that is more responsive to the strawberry polyphenols.
For future testing with ginger, I would start by removing the 2 antibiotics included in the media meant to protect it from microbiological organisms. I would also suggest the use of freshly picked ginger rather than pre-ground for spice use. Literature also calls for another media to be used, not the media that we have used in Julie's lab. In all of our experiments, ginger yielded positive results for killing leukemia cells. And because of that, there should be no major needs for changes in this experiment.